Hello everyone, my name is Alain Salvador Conejo Davila, and in representation of Dr. Armando Zaragoza, I will present the research with title Development of Silk Fibroin-g-PAni as Platform for Biocompatible Semiconductor Biosensors. But what is the fibroin? The fibroin is a protein present in silk. It is produced by diverse insects like larvae of Bombyx mori and other moth genera such as Anthrae, Circulia same, and Gotham. This protein is constituted mainly by glycine, alanine, serine, tyrosine, and valine. Fibroin, like a protein or biopolymer, presents great mechanical properties and flexibility. Also, is a biocompatible product with living organisms, present an environmental stability, and have a lot of functional groups in their structure which could be modified through chemical modifications. Due all these properties, fibroin was being employed in biomedical process to generate tissues, biological sensors, or gels. Polyaniline is a conjugated polymer with semiconductive properties, which presents three different oxidation states and could be doped or not. This variable determines the color and the conductivity of the polymer. Due to these properties, polyaniline was employed in developing colorimetric sensors, electrodes, supercapacitor, and transductors. The objective of this research is through a functionalization of fibroin graph change of polyaniline to develop a biocompatible platform with a semiconductor polymer which could be applied to generate new biosensors. The methodology starts cutting silk cocoons. After refluxing in sodium carbonate by 4 hours. This is to remove sericin, which cover the fibroin protein. To functionalize the fibroin, we use the alcohol functional group that is present in its structures, provided by serine and tyrosine amino acids. This amino acid will react with triamine benzoic acid in a Fischer esterification reaction. This reaction will produce an anilinium ion on the surface of fibroin, which could act as a graph point to produce polyaniline change. Once functionalized fibroin, it is solubilized in DMF and add anilinium salts to produce an oxidative polymerization in solution. This could graph PAni change to fibroin. To release the structural characterization of fibroin and their derivatives, we employed FTER spectroscopy. The spectrum of fibroin shows the three reported bands assigned to glycine, alanine, and serine amino acids, who are the main components of fibroin. After a esterification process, we can observe a white band, which is a sign of carbonyl of ester functional group and aromatic that substituent which comes from esterification process. Also, if fibroin-PAni spectrum we can observe the band of quinoid and benzoid ring. Those signals are characteristic of PAni formations, and by the intensity of those, we can deduce that is in leucomeraldine state. The bands of amino acids overlap with others, but the signal of serine is clearly identified in all spectrums. To complement the structural characterization, we realize and study through UV spectroscopy. We can observe for a fibroin functionalized a band at 2092, which is a p-transition of ring product by the functionalization of fibroin. We can observe after oxidative polymerization this sign decrease and appear two more signs, which correspond to benzoid ring and quinoid ring. Based on this, we can confirm the leucomeraldine state, but is undoped, or in other words, in not conductive form. To determine the thermal stability of fibroin and their derivatives, we employed thermogravimetric analysis. The degradation temperature for these three are so similar. It's between 200 degrees and 600 degrees. Nevertheless, we can observe that fibroin only presents two degradation steps. If we compare with esterification that presents three degradation steps, we can deduce that esterification medium, which is acid, degrade some the polymer change that present fibroin. Nevertheless, when we compare the thermograms with polyaniline fibroin that present polyaniline, we can observe only two degradation steps, and the second it's present a wide range compared with the other two. This is assigned to cross-link it between polyaniline change and fibroin fibers, which increase the thermal stability of the system. It's important in the system that present polyaniline determine the ratio between the components, because polyaniline is the main component of the system. The copolymer or the graph will present polyaniline properties, which are insoluble in solvents. It's a fragile compound and hard and so difficult to be processable. To this, we prepare 4 different graph systems, modify the relationship between functionalized fibroin and aniline salt. We can observe that benzoid signal starts to appear when aniline is almost at the half of weight formulation compared with fibroin. This indicates us that less than the half we cannot obtain polyaniline change. We only obtain oligomers, which are not interesting to develop a biosensor. This because their electrochemical properties are not good. Based on this, our final system, we need to have a composition of aniline salts almost at the relationship of the half base to functionalized fibrin. As a conclusion, through esterification of alcohol substituent in fibrin structure, we graft polyaniline change. The size of the PAni change is regulated by the condition of oxidative polymerization. The relationship between the fragments that compose the copolymer and the fundamental in the mechanical, thermical, conductive, and optical properties of the PAni, which will be studied in the next stage of the project. For our future work, we need to study the cytotoxicity of graft copolymers, also study the electrochemical activity of copolymers, and develop a flexible substrate with a conductive properties able to produce a biocompatible sensor. Thanks for your attention.